Okay, so today we're going to start chapter 4. This is the last chapter in this unit, and we'll see how many parts are going to come from it. So, chapter 4 is all about the cell. So, we're actually going to be diving into the cell, seeing what different kinds of cells there are, and seeing also the different features found in each of these two cells. Okay. Okay, so organisms are basically made out of cells. This is what the cell theory states. The cell theory is basically a theory that states that all living things are composed of cells. Now, these living things can be composed of one single cell, and that can constitute a living organism for some organisms, and and other organisms can be made out of multiple cells. So those would be a little bit more complex organisms like humans or like animals or plants. Okay, so it states that all living things are composed of cells and that all cells come from earlier cells as well. As we'll see in later chapters, and how I kind of mentioned, I believe, in chapter 1, a cell gives rise to another cell, to other cells. So a cell multiplies or divides, and this is how we have the continuity of cells as well. So organisms are either single-celled. These organisms include things like prokaryotes. Prokaryotes is a fancy way of saying, is basically a fancy category that includes things like bacteria and archaea. And it also includes certain protists. Protists are single-celled protists include things like single-celled algae and just funny little weird critters that we find in normal things like ponds and stuff. Or they can also be, organisms can also be multicellular, in which case they're plants, animals, and most fungi as well. So every cell in your body and every other living organism on Earth was formed by a division of a previously living cell. This is also what the cell theory states. So the cell is basically the fundamental unit of life. Okay, a life has to start with at least one cell. So again, you can have organisms that are single-celled, so that they have one cell and they constitute life because they're living and they're organisms, or you can have organisms that are made out of more than one cell and they are multicellular. Okay, but at the basis of all life, there's at least that fundamental unit, which is a cell. So the cell is the simplest collection of matter that can be alive. So many of forms exist as single cells. Many complex organisms and life forms exist as multicellular, so many cells. So cells can differ substantially from one another, but they share some common features. So as we'll see in upcoming slides, there's two categories of cells, okay, that we can have, that an organism can have. We can either have prokaryotic cells or eukaryotic cells. And so if an organism is a prokaryote, it only has prokaryotic cells. If an organism is part of the eukarya domain, it only has eukaryotic cells. And we'll see what the difference between these two cell types are. In order to visualize cells, cells are basically microscopic. They're too small for the eye, for the naked eye to see. So we actually require specific tools to see them. Usually these tools include things, include a microscope, right? So if you see here the scale, I'm just going to make it a little bit bigger. With this scale, you can see that frog eggs are usually about a centimeter, actually a little bit smaller than a centimeter in diameter. We have this paramecium is a kind of protist. It's just a weird single-celled organism that lives in the water. And then like actual cells tend to be smaller than 100 micrometers in length, right? So they're really, really small. And then of course, the actual features inside a cell, like nucleus or mitochondria, are even smaller. Viruses are the tiny, like extremely tiny entities, very, very tiny entities. And then of course we have the atom here, which is the smallest matter, right? The smallest unit of matter. So cells are very small, and in order to visualize them, we can't with the naked eye. If you see here, we require a light microscope to do so, a microscope of any kind really to do so. And there's varieties of different kinds of microscopes. So microscopes are used to visualize cells. Light microscope use visible light um, to actually see your specimen, whatever specimen you are looking at. So the visible light basically gets um, you put your specimen in your microscope, and then when you turn it on, the light goes through your specimen and into the different objectives and ocular pieces that then filter and tunnel the lights so that when you look at them through the binoculars of the light microscope, you're basically seeing a magnified image. So light microscopes, as their name suggests, use visible light and glass lenses to basically magnify your specimen. The specimen can be magnified up to about 1,000, even 2,000 times, depending on the actual light microscope. And so this way you can, you've magnified, you've zoomed into the image, and you can start seeing it a bit more. So this gives you, this is right here, a light microscope image of paramecium, which is that protist that I mentioned even in the past slide. This is what you normally see under an incredibly good, actually, this is a very good picture, like a very good visual of paramecium under the light microscope. So with light microscopes, you can see things that are both dead and living. So you can even, if it's living, you can even follow it around because it might even be swimming in, the various waters. Electron microscopes, as their name suggests, use electrons to actually magnify the image. Basically what they do, the specimen first has to be prepared. The specimen, when we look at specimens under the electron microscope, the specimen has to be dead. So we have to basically kill and fix the specimen and kind of cover it with a specific heavy metal, so something like gold or platinum has to cover like a very thin layer of gold has to cover our specimen, and then we shoot electron beams at it, and whatever the electrons are going to then bounce off of our specimen, and that pattern, the scatter pattern that we get once the electrons like bounce off and kind of are reflected back, you can imagine, or reflected back at you, that's going to create a magnification. And this magnification is incredibly potent, has a very high magnification of up to like 1,000,000 times. And in addition, the resolution, so basically how clear the image is remarkable. So it can magnify things about 100 times more than a light microscope. So visualize things that are even 100 times smaller than what light microscopes can normally, like compared to the specimens that light microscopes can normally see. And when they do so, they do it in very clear detail. If you can see here, you can see all the little hairs. These hairs are known as cilia, okay? All of these little hairs basically found on the paramecium. It's very incredible detail. And you can also basically zoom in. These are the same hairs that we saw in the past in this picture right here, right? You can go ahead and magnify even more so that you can see the individual hairs and basically how they're stuck to the cell, right? It's incredible, the actual, the resolution and the potency of the magnification of electron microscopes. This, however, doesn't mean that light microscopes, so even though electron microscopes have better clarity, basically better resolution, and better magnification than light microscopes, this doesn't necessarily mean that light microscopes are useless. Because with electron microscopes, we can only see dead organisms. A lot of times we want to study our specimen in its environment and we want to see what it's doing. And so light microscopes are much handier in that case, because light microscopes, all you're doing is you're putting light through a specimen. The light is not going to kill your organisms, and so your organisms can be alive while you visualize them under a light microscope. So each microscope has its own advantage. Another thing to note is that light microscopes are a lot cheaper than electron microscopes. Electron microscopes tend to be very bulky and big, and you need incredibly specific software and programs, and even its own computer basically to visualize the images projected by the by the electron microscope. So let's look at the 3 domains of life, because as we look at the 3 domains of life, we can see which domain has which kind of like cell type. So the 3 domains of life are bacteria, okay, archaea, and eukaryotes. And then the eukarya or eukaryota or eukaryotes, as long as it's eukarya something, this domain can then be split into 4 kingdoms: animals, right, fungi, plants, and protists. So we have the eukarya domain, which includes a lot of different kinds of organisms, bacteria, and archaea. The basic structural and functional unit of every organism is a cell, and we have 2 types of cells. We have prokaryotic cells and eukaryotic cells. You're going to need to know the difference between these cells. This is critical for basically not just this unit, but all units, after this. So for the rest of the semester. Prokaryotic cells are cells that can be found in the bacteria domain and the archaea domain. So if we find a bacteria, okay, which is a single cell, that cell is going to be what type of cell? A prokaryotic cell. Okay, if we find an archaea, that cell is also going to be a prokaryotic cell. Eukaryotic cells are found in the eukarya domain. That's why we're called the eukarya domain, because we have eukaryotic cells. So protists, fungi, animals, plants, we all have eukaryotic cells that make us. You don't, even though you're a multicellular organism, you don't have any prokaryotic cells in you whatsoever. All of your cells are eukaryotic cells. Same goes for plants and fungi and other animals and protists. They all have eukaryotic cells. Bacteria are not made out of eukaryotic cells, nor are archaea. They're made, both bacteria and archaea, their cells are prokaryotic. So what does it mean to be a prokaryotic cell or a eukaryotic cell? This table right here kind of, this table right here summarizes it very well. So prokaryotic cells tend to be smaller than eukaryotic cells. Eukaryotic cells tend to be a little bit more big, okay? Another thing about prokaryotic cells is that they're a lot simpler. They're very simple. Basically, they have the, here we go. Prokaryotic cells are a lot simpler. They have this jelly-like fluid encompassed within the actual cell. So they have a boundary around the cell. Like when you typically draw a cell, you start by drawing a circle. That circle is the boundary, and it's known as the cell membrane or also the plasma membrane. I may interchange those two terms quite frequently. So the cell membrane or the plasma membrane is going to be your barrier, your boundary that actually encompasses the cell. And then within that boundary, inside of it, we have this jelly-like fluid. It's kind of like a viscous jelly-like fluid known as the cytosol. And now, basically, that's the main part of the prokaryotic cell. Everything else that is found within the prokaryotic cell is just swimming and floating around, suspended within this cytosol jelly-like fluid. Okay, the cytoplasm is just the area where the cytosol is found. So if I say cytoplasm or cytosol, or if you say cytoplasm or cytosol, it usually means the same thing, right? Cytosol is just the fluid. Cytoplasm is the area where we find that fluid. It's the region where we find that fluid. So inside the cytosol or inside the cytoplasm, we have whatever kind of proteins, any of the other macromolecules that we looked at, the DNA, the RNA, proteins, carbohydrates, lipids, they're just found suspended within the cytoplasm, just kind of swimming around. But they're not put in any weird little compartments. They're just found basically floating in it. It's kind of like an open floor plan, right? Imagine having like a studio apartment where you have your bedroom and your kitchen in the same enclosed space. That's what's happening in a prokaryote. You don't have a room for your bedroom. You don't have a room for your kitchen. Everything is basic. It's like an open floor plan. Everything is out in the open. Okay, so they're again generally really small and quite simple. Prokaryotic cells, any organism that is made out of prokaryotic cells is always only single celled. So a bacteria is single celled, archaea is single celled, and cell is a prokaryotic cell. And even though they don't look like there's a lot of variety in them, that there's a lot of stuff happening, there actually is metabolically, so their chemical reactions, they're really different from each other. And so there's a lot of variety in different bacteria and archaea, even though morphologically, just like looking at them right now, they look very similar. Like you can't really see that much difference, right? Okay, so just to go over a couple of features that, so that's a prokaryotic cell, sorry. Eukaryotic cells, let's go back. Before I go over the features that they have in common, eukaryotic cells are usually larger. And then if you look here, it looks a lot more complicated than your small little prokaryotic cell, right? Here we had the open plan, and all of a sudden inside the eukaryotic cell, we start seeing compartments, so we start seeing rooms, okay? This is because eukaryotic cells have compartments. These compartments are known as organelles. So you can kind of think of them as little organs for your cell, okay? Inside the eukaryotic cell, we have organelles, and in these, or each organelle does its own thing. So we have, for example, the nucleus, and the nucleus holds the DNA of the eukaryotic cell. Well, in prokaryotic cells, the nucleus was just lying around, okay? It was just lying around in the cytoplasm. So we have membrane-bound organelles in the eukaryotic cell. So things like the nucleus, the mitochondria, the endoplasmic reticulum, the Golgi apparatus. If you don't know what these words are, don't worry. We're going to go into more detail on each one later on in the chapter. But basically, this is the main difference between eukaryotes and prokaryotic cells. Prokaryotic cells, they do not have membrane-bound organelles. They don't have little compartments within them. And eukaryotic cells do have membrane-bound organelles, and so you can start seeing these weird compartments. In fact, actually, if you you can try to remember, if you have difficulties remembering which one's which, you can look at the actual word. So karyon in Greek, karyo comes from the word karyon in Greek. Okay, if you see here prokaryo and then eukaryo right here. Karyon in Greek means like kernel or the center or basically the nucleus of something. Okay, like the center, the middle of something, where everything gets programmed. Prokaryote then therefore means before the nucleus. So if it came before the nucleus, it doesn't have a nucleus. And eu means true. So eukaryote means true nucleus. So prokaryotic cells came before the nucleus and they do not have the nucleus or any other membrane-bound organelles. Eukaryotic cells have a true nucleus and therefore have also other membrane-bound organelles. So this is a major difference between prokaryotic cells and eukaryotic cells. They also have a couple of things in common. So basic features of all cells. All of them have a border. All of them have a plasma membrane or a cell membrane. So I'm going to write here, also known as a cell membrane. Okay. And this cell membrane is a selective barrier that allows specific things to come inside and specific things to go outside. It basically keeps things in, right? Keeps all the stuff that's found within a cell inside enclosed in an area, and then it allows certain like nutrients to filter inside and waste to go outside. It allows oxygen to come inside and different kinds of excrements to go outside of the cell. So it chooses basically what goes in and out. It's very selective in that sense. You also have a semi-fluid, that jelly-like substance called a cytosol. So the cytosol is a semi-fluid jelly-like substance in which all subcellular components are suspended. The area where we find the cytosol, so the region of area where we find the cytosol is called the cytoplasm. Okay, so usually the cytoplasm is any region that is enclosed by the plasma membrane that doesn't include the nucleus. They also have chromosomes. Chromosomes is just where we have our DNA. They carry our genes. So this is, the chromosomes carry our genes, and this is basically DNA. So every single cell has DNA. And they also have other, like these little organelles called ribosomes. So yes, prokaryotes have organelles called ribosomes, but ribosomes are not membrane bound. So they're not a compartment. They're basically bigger, they're basically composed of RNA and protein. So they're not actual they're not actual fully fledged membrane bound organelles. So again, prokaryotes do not have membrane bound organelles, but they do have these things known as ribosomes, which are kind of like big little things, right? And they're really important. Ribosomes are really critical because they help make proteins. We'll talk about ribosomes in just a sec. Okay. So those are the features that are found the same in prokaryotes and eukaryotic cells. And then, of course, now we can look into prokaryotic cells a little bit deeper that we were doing before and see even more, right, what there is. So again, there are no membrane-bound organelles. We do have ribosomes in them, but there's no membrane-bound organelle. So we don't have a nucleus. We don't have a mitochondria. We don't have an endoplasmic reticulum. We don't have a lysosome. None of that, right? Our DNA, the DNA in prokaryotic cells, isn't just found basically floating like a bunch of spaghetti all over the cell. It's concentrated in one specific region. So if you see here, this blue thing right here, this is where the DNA is concentrated. It's not bound by anything. There's no compartment around it, but the DNA does stay in that specific area. This area is known as a nucleoid. So don't get this confused with nucleus. It sounds very similar to nucleus, right? Don't get that confused with nucleus. The nucleoid is an unbound region where the DNA of prokaryotes, so bacteria or archaea, is found. different from a nucleus. A nucleus has an actual membrane around it. Okay, so it's an actual container that contains DNA. Nucleoid is just a region, it's just like a field. Okay, so they don't have any membrane-bound organelles. They have a cytoplasm, and there's all this stuff that's found basically suspended in the cytoplasm, like other proteins and everything. So it's not actually formless, even though it looks formless under the light microscope. Prokaryotes are very, very tiny, so bacteria or archaea are very small. This is a head of a pin, and it's magnified 85 times. And you can start seeing the bacteria just a little bit. If I zoom into 425, you can see the bacteria even more. And then this is a magnification of 2,100. Here we can actually see the actual bacteria, right? So they're very, very tiny, and they're incredibly numerous. Other things that prokaryotes can have, they can have a couple of things kind of like projecting out of them, like sticking out of them. They can have things known as fimbriae. Fimbriae are little sticky thistles. You can kind of imagine them as little thistles on the bacteria. Some bacteria don't, or some bacteria or archaea don't have them, and some bacteria do. So it depends on the actual species of bacteria we're talking about. So fimbriae are these little thistles that help kind of attach bacteria to other things. Um, so this is kind of awkward to even talk about, but Neisseria gonorrhea, which is the bacteria that causes gonorrhea, the reason why it's sexually transmitted is because the thistles, these fimbriae, allow the bacteria to basically hitch a ride with the sperm during sexual intercourse. So that's something to think about. A lot of bacteria also have a flagellum, okay? Sometimes they have one flagellum, sometimes they have multiple flagella. So flagella is a plural of flagellum. The flagellum is basically this tail whip, like this little whip, this little tail-like whip that allows for movement of the bacteria. Okay, so fimbriae allow for attach, they're just sticky, they allow the bacteria to attach, and the flagella allows for movement of the bacteria. So literally the bacteria can swim, okay? And actually the flagellum in bacteria spins like a propeller, and that's how the bacteria basically move through their environment. A lot of the times they all have a plasma membrane. So we've looked at the plasma membrane. It's that initial boundary, right, or the cell membrane. Outside of the plasma membrane, most prokaryotic cells also have an additional barrier, an additional kind of layer of protection known as the cell wall. Which is really important for bacteria, right? A lot of times, many antibiotics that we have that we take to get rid of bacteria, some of these antibiotics just try and destroy the cell wall of bacteria. So this is good because we, as animals, our own eukaryotic cells do not have a cell wall. Animal eukaryotic cells do not have a cell wall. And so the antibiotic is specific to target cell walls. And so it's not going to harm our own cells, but it will harm bacterial cells. So that's good. Sometimes they can have even more protection and have a capsule. So even like on top of a plasma membrane, on top of a cell wall, they have a capsule. So these capsules make the bacteria very hard to get rid of, and this can make the bacteria actually pathogenic and cause diseases because we have difficulties getting rid of bacteria that have capsules because they're just so protected by all of these layers, right? Inside of the bacteria, we have this jelly-like fluid called the cytosol. The region, the area where the cytosol is found is known as the cytoplasm. Within the cytoplasm, we have different things suspended in it. So whatever enzymes and proteins and carbohydrates that the bacteria needs to survive are found in here. We also have ribosomes, which are the site of protein synthesis. This is where we actually make, the bacteria actually make their proteins. And then the DNA, because every single living organism has DNA, is found, remember, in that region, that concentrated region known as the nucleoid, not the nucleus, the nucleoid, because the nucleoid is unbound. It doesn't have any membrane that's basically constraining it or binding it. Okay, so then we move on to eukaryotic cells. Just really quickly, the eukaryotic cells are characterized by having DNA in a nucleus. Remember, true karyon, true nucleus, eukaryote. They have a nucleus. So inside this nucleus is this purple thing or pink purple thing right here. Inside the nucleus, we have the DNA. There's also other membrane-bound organelles. So the nucleus is membrane-bound. As you can see, there's like an actual layer around it, okay, that encloses the DNA. And then there's other weird little compartments in here. All of these are membrane-bound organelles, okay? So the cytoplasm in this case is the region between the plasma membrane and specialized organelles. So wherever we find the cytosol is where we have the cytoplasm. Eukaryotic cells are generally much larger than prokaryotic cells. They're usually about 10 to 100 micrometers in diameter, and prokaryotes are about a tenth of the length of a typical eukaryotic cell. Actually, if you look at a eukaryotic cell, I know this isn't obviously to scale, but the size of a eukaryotic cell, the size of the nucleus of the eukaryotic cell is usually how big a prokaryotic cell is. So if I were to draw a eukaryotic cell, right, I'd start off with drawing my plasma membrane or my cell membrane, and then I would draw, for now, just my nucleus. I'm going to label it because this is what you do in biology. You label everything, right? You label the nucleus. I'm going to write here plasma membrane. So this is obviously, even if I didn't tell you this was a eukaryotic cell, you would be able to say that it was a eukaryotic cell because it has a nucleus. So I'm going to write here eukaryotic cell, euc. Okay. A prokaryotic cell, if I was to draw this to scale, would basically be about this size, right? And you can see how it's a size that is comparable to simply the nucleus of the cell. So prokaryotic cells, a lot smaller. They also have a plasma membrane, but they don't have a nucleus. In fact, their DNA is just over there, kind of floating around in that area, the region called the nucleoid region. Okay, they don't have a membrane. If I was to draw a membrane around this, if I was to do this, see how now it's enclosed? All of a sudden, this would not be a prokaryote. This would be a eukaryotic cell. Okay, so prokaryotic cells don't have any membrane-bound organelles. They don't have any compartments. All their stuff is just floating around within the cytoplasm like so. While in eukaryotes, we have all sorts of things that are compartments that are found within the actual cell. Okay. So I think this is where I'm going to stop for now, and then in the next parts of the actual chapter, we're going to go in and actually look at the eukaryotic cells and what makes up like the different features of the eukaryotic cells, and just generally different features of all other things as well. Okay, so I'll see you guys in the next video.